Welcome to Computational Biology. Today, we are diving into the fascinating world of gene and protein sequencing. Imagine you are handed a massive, ancient library filled with books, but all the books are written in a secret code. You know they contain the instructions for building a human body, but you cannot read them yet. Sequencing is the ultimate code breaker that allows us to read these biological letters one by one, unlocking the very instructions of life. Building on that library analogy, let us look at the alphabets used in these biological books. Why does this matter? Because before we can analyze data computationally, we must understand its physical form. Instead of English letters, genes are written in a 4-letter alphabet of chemical bases. Proteins, on the other hand, are written in a 20-letter alphabet of amino acids. Sequencing is the incredible technology that allows us to read these biological letters in their exact order, transforming physical molecules into digital text strings that we can study. Now that we know the alphabets, what exactly is sequencing in a laboratory setting? In biochemistry, sequencing refers to the precise methods used to determine the exact sequence of monomers in a biological polymer. For DNA, this means finding the linear order of adenine, thymine, cytosine, and guanine. For proteins, it involves determining the specific arrangement of amino acids linked by peptide bonds. This is crucial because this primary linear structure dictates how the molecule will fold into a 3-dimensional shape, which ultimately determines its biological function in the cell. Understanding this primary structure leads us to a critical question. Why does reading this biological text matter so much to us? The sequence of these molecules holds the key to understanding health, disease, and evolution. If even a single letter in a gene sequence is mutated, it can cause severe genetic disorders like sickle cell anemia or cystic fibrosis. In the real world, sequencing is the backbone of personalized medicine, allowing doctors to tailor treatments to a patient's specific tumor. It also allows scientists to track viral outbreaks and develop vaccines rapidly. Without it, we would be completely blind to the molecular causes of disease. To truly grasp this transformation, let us look at this visual representation of the sequencing process. On the left, you see a physical DNA double helix being processed by a sequencing machine, which unzips and reads the bases, outputting a digital text string of A, T, C, and G. On the right, a complex folded protein molecule undergoes a similar analysis, resulting in a digital sequence of amino acids like methionine and valine. The central arrows highlight the core concept. We are taking physical biological samples and transforming them into digital sequence data. This digital output is what computational biologists actually analyze. Having seen how we get this digital data, we must now understand the sheer scale of it mathematically. Where does this vast diversity of life come from? It comes from combinations. The formula n equals a to the power of L calculates the total number of unique sequences that can possibly exist. Here, n is the total number of unique sequences. The variable a represents the size of the alphabet, which is 4 for DNA and 20 for proteins. The variable L represents the length of the sequence. Physically, this means that if the sequence length L increases even slightly, the total number of possibilities, N, explodes exponentially, allowing for massive biological diversity from just a few basic building blocks. Let us apply this formula to a concrete example to see how it works in practice. Suppose you are studying a very short DNA sequence that is exactly 5 units long, and you want to know how many unique variations could exist. We are given the length L equals 5. Because it is DNA, we know the alphabet size, A, equals 4. We chose our exponential formula because we want total combinations. Substituting these values, we get n equals 4 to the power of 5. This means choosing one of 4 bases for each of the 5 positions. Calculating this out gives us our final answer. There are 1,024 possible unique DNA sequences of length 5. Now, let us compare that DNA result with a protein of the exact same length. We are still looking at a sequence length L of 5. However, for proteins, the alphabet size A is 20 because there are 20 standard amino acids. Using the same formula, we substitute A equals 20 and L equals 5, giving us 20 to the power of 5. When we multiply 20 by itself 5 times, the result is drastically different. The final answer is 3,200,000 possible unique protein sequences. Physically, this shows that proteins have a vastly larger vocabulary than DNA for the exact same length, enabling them to form highly complex structures to perform cellular work. These massive numbers of combinations bring us to an important concept in computational biology: probability. When we compare digital sequences from 2 different organisms and find a long, identical match, we have to ask, did this happen by random chance? Because the number of possible combinations is so astronomically high, the probability of finding an exact long sequence by pure chance is incredibly low. Therefore, when we do find these matches, it strongly tells us that the organisms share an evolutionary ancestry. To prove this mathematically, we need a way to calculate the exact probability of a specific sequence occurring randomly. This brings us to the mathematical formula for sequence probability. Conceptually, this formula derives from multiplying the independent probabilities of getting a specific letter at each position in the chain. The formula is P equals 1 over A, all raised to the power of L. Here, P represents the probability of a specific sequence occurring purely by random chance. The variable A is the alphabet size, and L is the sequence length. If the sequence length L increases, the fraction gets multiplied by itself more times, meaning the probability P decreases dramatically. This mathematical relationship is what allows us to confidently identify non-random, biologically significant patterns. Before we calculate our next example, let us understand the biological tool we will be using. In the laboratory, scientists use proteins called restriction enzymes, which act like molecular scissors. These enzymes scan along a long DNA molecule and cut it only when they find a very specific sequence of letters. For instance, an enzyme called EcoRI cuts DNA exclusively at the exact 6-letter sequence GAATTC. Because genomes are massive, we use probability to predict mathematically how often these specific cut sites will appear by random chance, which helps us plan our experiments. Let us see how this probability formula guides real-world laboratory experiments. We're given a restriction enzyme called EcoR1 that cuts DNA only at the exact sequence GAATTC. We are asked to find the probability of this sequence appearing by chance in a random DNA genome. The sequence length L is 6, and the DNA alphabet size A is 4. We choose our probability formula and substitute these values, giving us 1/4 to the power of 6. This means at each of the 6 positions, there is a 1 in 4 chance of getting the correct letter. Multiplying this out gives 1 over 4,096. Our final answer means you would expect to see this cut site roughly once every 4,096 base pairs, helping biologists predict how a genome will be chopped up. We have covered a lot of ground today, so let us summarize the key takeaways. First, gene and protein sequencing is the foundational technology that acts as a bridge, converting physical biological molecules into digital data that we can analyze. Second, we learned that while the biological alphabets are small, just 4 letters for DNA and 20 for proteins, the number of possible combinations grows exponentially as the sequence gets longer. Finally, we saw how the mathematics of probability allow us to distinguish between random chance and true evolutionary ancestry. Mastering these concepts is your first step toward understanding how computational tools unlock the hidden libraries of life.